Okay, so today we're going to be looking at a different form of the cardiac cycle, the pressure volume loop. So if we have some sort of graph with pressure here, and volume down here, and we can make a graph of the chambers of the heart, the ventricle, and what it does during the cardiac cycle. So we have here, we'll put 50 milliliters. Up here is about 120 milliliters. And then pressure, up here we have 120, and right here we have 80. And down here we have like 10, it's not very pressurized. But, so what happens is, we have a loop that goes kind of like that. I'll move this over here. That's what the pressure volume loop looks like. And each one of these parts is a different reason, there's a different purpose for it. So this right here, this is the ventricle filling up. And so what we get here is the ventricles going from 50 milliliters of blood to about 120. So we're really filling it up and then we're pumping it out. And this point right here, at the very bottom right-hand corner, this is our preload. And that's just the amount of blood that's in the ventricle right before it starts its isovolumic contraction, and we start getting the heart contraction cycling. So this is also where the pressure builds up enough to pump, and we get that isovolumic contraction until the aortic valve opens and then it closes here. So we have the mitral valve opening and the mitral valve closing when we hit our preload, and then isovolumic contraction. And then here, I'll make this in green. Right here, that corner, we have what's called the afterload. An afterload, this represents all of the pressure on the ventricle that it needs to overcome to pump the blood out into the body. So this can be like vasoconstriction or your blood pressure, clots, the blood viscosity, things that will stop the blood from pumping through the system. And then there's one other point here that's really important. I'll make this one in black, is right here. There's a line right there. And this line right here, it represents the entropy. Inotropy. So inotropy it's how stiff the ventricle gets at maximum contraction. So it's kind of how hard the actin and myosin grab each other and really crank down to pump all the blood out. So with a higher entropy, we're going to get a stiffer ventricle at max contraction, and with a lesser entropy, we get a lower one. And that can be changed due to facts and things like stiffness of the heart, the thickness of the heart walls, and all of that stuff. So right here we have our end systolic. So this 50 or whatever is this lower end is that's left in that atria, that ventricle when it's done. This is the end end systolic volume. And this over here, the 120, where we hit our preload, this is the end diastolic volume. And that's important because right here in the middle, from the end systolic to the end diastolic, this area right here, this has a name. This is called the stroke volume. We'll put SV there. So stroke volume is how much blood is pumped out of the heart in one cardiac cycle. So we take our end diastolic and we subtract our end systolic from it, we get our stroke volume, how much blood is pumped out in this cardiac cycle. And that's very important because this can change. If we change our preload and we put it somewhere out here, we're able to get more blood into the heart, but everything else stays the same, the afterload stays the same. This would look more like that. We'd still get the same entropy, but we would have a higher stroke volume there. More blood would be pumped. And it's the same thing if we were to decrease it. We could get decreased preload, we could get a smaller stroke volume. We could also increase and decrease afterload, and that would change the height of it. So if we decreased afterload, I'll just do this one in green, these dots. We would come up here and then come across there. And that, if we decrease our afterload, we can actually change our inotropy. We can take this line and point it here somewhere, and we can actually change, I'll do it the other way. We could change the slope of that line and change our inotropy based off things like afterload. Stroke volume doesn't affect it too much. It can, but it doesn't necessarily. And then our different volumes can do it. Calcium can do it, the strength of it. So if we have like a higher pressure, a higher blood pressure coming out, then that entropy line can be changed. So inotropy line. The next thing we're going to talk about is blood pressure regulation. So blood pressure regulation, we have short-term and long-term. Today we're going to focus on the baroreceptor or the pressure receptor. So in our aorta, we have a heart here. We have intraventricular, the septum, the atria separated from the ventricle, and we have our aorta coming off here, coming out there. We're just going to focus on that aorta. So coming off the aorta, we also have other major arteries, and the big ones are our carotid arteries. So I'm just going to draw two arteries coming off here. So in our aorta, and these carotid sinuses, we have some receptors. These are our baroreceptors. What they do is they are a pressure receptor. They sense changes in pressure. And what they'll do is they will actually send potentials to the brainstem. So we have the brainstem over here with the sympathetic and parasympathetic nervous system in it. We're going to do that in green. Nerves are in green. And they'll come over and they'll kind of synapse with both of those. And this is important because if we have increased pressure in the aorta, we're going to send a higher frequency of action potentials. We get more excited. It's in a more excited state. So the frequency would go, our baroreceptor stimulation goes up, the action potentials, I'm going to put APF, action potential frequency, would go up. And what does that tell the brainstem? It says, hey, we have a high blood pressure. There's a lot of pressure against these baroreceptors. So it's going to come over, it's going to activate the parasympathetic nervous system, which will send acetylcholine to muscarinic receptors to slow down the heart, to vasodilate, to do all these things to help lower that blood pressure. But what if the opposite happens? If the opposite happens and our blood pressure drops, the baroreceptors, they won't stretch as much, so they won't be activated as much, and we get a lower action potential frequency. So the blood pressure drops momentarily, the baroreceptors don't get stimulated as much, fewer action potentials. The sympathetic nervous system needs to be activated, so it'll synapse with the sympathetic nervous system. Sympathetic nervous system will send out the epinephrine and norepinephrine and activate the beta receptors, alpha receptors, vasoconstrict, pump more blood, the heart rate speeds up, stuff like that, to try and compensate for this, to make up for it. And that brings us to our next point. So our arterial pressure, our blood pressure, it's in a constant state of change. So we usually stay between 120 and 80 in our big arteries. So we have our arteries, and we'll stay kind of between those. But as our arteries go to arterioles and then the capillaries and venules and veins, this pressure kind of dips down and it just kind of does that sort of thing. So our arteries, we have arterioles, capillaries, there'll probably be more over here, venules, and veins. Veins don't have a lot of pressure. They're probably sitting here at around 5 to 10. So we'll put 10 here because that's what the capillaries and arterioles and venules, that's what is kind of most important to them. But if we were to draw kind of like a line of best fit through here, we would get that line. It kind of cut through these fluctuations and then follow that line down. This is what we call our mean arterial pressure. Or MAP for short. So this can be found in a couple different ways, but it's important to understand that the average pressure, or the average arterial pressure, if we were to take it, it's going to fall somewhere on that line as we go throughout, depending on if we're taking in arteries, arterioles, capillaries, veins, or venules. It's going to fall somewhere on that line. And the way we maintain this is things like heart rate, vasoconstriction, the things that we do to maintain blood pressure. We can maintain this mean arterial pressure that same way. And then like entropy, inotropy, that kind of stuff to really just push the blood out, and that's how we affect this mean arterial pressure. And we want to keep that, it falls closer to the diastole pressure than it does the systole pressure, and that's a good thing. We want it to do that. And it's because at any given point in time, your body's usually in diastole. You have spent more time in diastole than systole, and so that's naturally where it's going to fall. So the last thing we need to talk about is capillary exchange. So I'm going to draw a little thing here. Okay, this is a capillary, a single capillary. We're zoomed in, major time. And over here, we have some arterioles. The arteries come to arterioles, down to the capillaries. we have some smooth muscle here. They call it a sphincter. It's not really a sphincter, but it will constrict to not let blood flow into the capillaries if we don't need it in there. If we're trying to increase blood pressure and vasoconstrict and control what's happening in the capillaries, this is what does it. They will constrict and not allow blood cells to get in here. Because this capillary, it's big enough for one red blood cell to kind of go through here at a time. So the red blood cells are kind of going through here single file and doing fluid exchange. And then we're going to talk about what causes fluid exchange. So there are 4 pressures that we need to keep track of in the capillary. The first one is hydrostatic pressure. The second one is what's called BCOP, or blood colloid osmotic pressure. So BCOP. Then we have ICOP, or interstitial colloid osmotic pressure. And then the very last one we're going to talk about is just interstitial fluid pressure, so IFP. Okay, now let's just change this to HP. Okay, so first thing, hydrostatic pressure. This is the pressure that the blood cells, whatever it is, puts against the walls of the capillary. So we have our hydrostatic pressure that is pushing pressure to the outside. We have our BCOP, which is an osmotic pressure, and because there's a lot of proteins, albumin, big proteins that won't cross this layer of cells, that's going to be an inward pressure. Then we have our ICOP, and this is colloids and proteins on the outside, so that's going to be an osmotic gradient pulling water or fluid out. Then we have our IFP, interstitial fluid pressure, and this is pressure that's generated by the lymphatic system that pulls fluid out of the capillary. It's a pretty small force, but it does do so. And this is where things get interesting. I'm going to draw this one in green. We have another hydrostatic pressure here, because as you go across the capillary, the hydrostatic pressure changes as you go through. And that is why at the arterial end, fluid is being taken out and it's oxygenating cells, it's delivering nutrients to cells and stuff. But on the venule and vein side, it's pulling fluid back in to go get oxygenated nutrients and stuff like that. And this is kind of how this works. So if we were to give these numbers, your IFP, we'll start over here on the vein side. The hydrostatic pressure on the vein side, this is about 10 millimeters of mercury. And then the interstitial fluid pressure, this is about 3 millimeters of mercury. The ICOP here, it's about 8 millimeters of mercury. BCOP here is 30 millimeters of mercury, and hydrostatic pressure, it's about 28 millimeters of mercury, somewhere around there. So the reason that this is super important, oh, actually I got these switched. Hydrostatic pressure here is 28, is 30. This one's 28. Okay. So what happens is we have a red blood cell come in. It exerts this hydrostatic pressure, and we have fluid in here. We have this fluid in here that's trying to get out and get to the interstitial space, the cells out here, like little cells there. So if we want to know which way fluid is going to flow, we can make a little chart. We have pressure in and pressure out. So pressure in, if we look at all of these right here, we only have beak off that's actively having pressure to bring stuff in. So we can put 28 millimeters of mercury there on the arterial side, but then pressure out, we have 30 here. And then we have 8 and 3. So in all, we would have 28 in and 41 out. So we would have a net outflow of fluid. There's pressure pushing it out. And it's only like 13 millimeters of mercury. It's not a ton of pressure, but it's enough to get it out. However, if we were to look at the vein side of things, we now have pressure in stays at 28. ICOP doesn't change. IFP doesn't change. However, hydrostatic pressure, that drops down to 10. So now if we were to do this, we still have 28 over here, but over here we have 21. So that would mean there's a 7 millimeters of mercury inflow. So we're bringing fluid into the cell, into the bloodstream to be taken back out into the body. And that's really what happens in the capillary. These are the important pressures in the capillary, and this is what makes fluid exchange possible. So, yeah, and as I explained this, hydrostatic pressure caused by fluid. This is called by proteins in the blood, proteins in the interstitial fluid, and this is called by lymphatic vessels that are just kind of pulling a little bit of pressure out. So that brings the question of swelling, edema. What happens? So let's take right-sided heart failure for an example. We have our inferior vena cava, right? If we're looking at a capillary in the leg, we have our inferior vena cava that's pulling blood up into the right side of the heart. If the ventricle on the right side of the heart is swollen or too thick and it doesn't have a big enough volume, we can't put enough blood in it. So blood will not go into the ventricle, will stay in the atrium, then the atrium will back up and it'll keep kind of a pressure of blood down the inferior vena cava back into the legs. So we have a little bit of pressure coming back this way, which would increase this vein side hydrostatic pressure. So over time, we say this can get up to, let's say, 20 millimeters of mercury. That's a big jump. That's like some major right-sided heart failure. We're getting a real big jump, but just to prove my point. So we have pressure in and pressure out. So our pressure in, if we're looking at the vein side now, our pressure in is 28 from this beacon. But if we look at pressure out, we have 8, 3, and now 20. Instead of 10, it's 20 now. So if we were to add this up, we have 31 millimeters of mercury of pressure here and just 28 millimeters of mercury here. So that means that the vein side is no longer going to be pulling fluid in to send it back out to the veins. We have more pressure here flowing out into the tissue than flowing into the capillary, making us swell. We're going to send fluid and other things out into the cells, causing swelling. And other things can cause this. Lymphatic problems can cause this. If we lose lymphatics and they're no longer pulling, we can lose this 3 and it can go to 0 and change things. And it's just, there's a lot of different things that will change these pressures. And I believe that is all we have to talk about today.